All right, everybody, welcome to our very special educational, informational session this evening. My name is Lori Clark, and I am very honored and very privileged to be able to host an incredible guest, an incredible individual with information that everybody, no matter what your age or your stage of life is interested in learning about. And so we are going to, on this Wednesday, March 16th, 2016, offer the opportunity for all of us to learn and be educated on the quest for immortality. And Dr. Ruggiero is an absolutely beautiful, heart-centered human being who has been studying and researching this very topic for many, many years. And what I want to do first, before we do anything, is just give you a bit of background on who this amazing individual is. And the gift of his time for us tonight is so graciously appreciated. He was born in Firenze, Italy in 1956. He graduated from the School of Medicine of the University of Firenze in 1980. He has served in the Army as Lieutenant Medical Officer. In 1984 to 86, he worked at the Laboratory of Cellular and Molecular Biology of Burroughs Wellcome Co., where he published a paper sponsored by Nobel laureate Sir John Vane. Take note of what I'm saying. We're speaking of an individual who's devoted his life to this area of research that you're going to learn about tonight. He subsequently worked at the National Cancer Institute of the National Institutes of Health for the USA in Bethesda, Maryland. So he had been in the US prior. He performed research on oncogenes and signal transduction. He returned to Italy as professor of molecular biology at the University of Firenze until his retirement in 2014. So in 36 years of this scientific career, Dr. Ruggiero has published more than 150 peer-reviewed articles, and he has been invited to participate to hundreds and hundreds of congresses and conferences to share his research and his knowledge and his gift. His main research areas of interest are in the fields of oncology, neuroscience, and immunotherapy. And he and his wife have recently relocated to Arizona, which is where they are today and where he is guest speaking and providing this information for us. And we are blessed with his knowledge, and just who doesn't nowadays want to know how to age well? Who doesn't want to understand the possibility of living forever? And what if you and the people you know and love could find a way to make that happen? What if it actually was true that immortality is and can be a possibility? Would you want to know a little bit more? And with that, I would love to turn the floor over right now. Get your notepads out, get your paper out. Dr. Ruggiero, thank you so much for your time this evening. I am beyond excited because I know a little tiny bit of tidbits of things you're going to be sharing with us, and I can't wait to hear the whole story.
Well, thank you so much, Lori. Let me show my full appreciation for you, for Diana and Caroline, and for all your community. Even though I don't know all the members of your community, I feel that I have a spectacular audience tonight. So I thank you so much for this great opportunity. So tonight we will explore this sort of biological, scientific, or even metaphysical topic that is the quest for immortality. But before we start, as always, a disclaimer. The following are academic and scientific considerations. They are not medical prescriptions or advices. Now, there is a sort of irony. Usually in the other phrase I write, please notice none of the information that will be presented is to be interpreted as a cure. And usually I say as a cure for a disease. But here the cure is for human mortality. So today we are talking about our condition of humans that we think that death is inescapable. So let's see whether it is truly inescapable or not. All right, so let's start with this article published in French in the Revue Médicale Suisse by a French author just a few months ago in June of 2015, dealing with immortality, death, or other minor phenomena. And actually, when we're dealing with immortality and death, all the other phenomena, including La Révolution Française, as this plaque in Grenoble says, become rather minor. Now, why did I decide to talk about immortality tonight? For the very simple reason that a few days ago, an article authored by myself and Mr. John Anderson, whom I think you know very well, was published in the very prestigious Encyclopedia of Cancer. The Encyclopedia of Cancer is published by Springer Verlag in Berlin and Heidelberg in Germany. And this article of ours deals with a gene that is difficult to pronounce. It is called BCL2. And this gene is also dubbed as the gene for immortality. Now, let me talk a little bit about the Encyclopedia of Cancer, because maybe you're not aware of this publication. Usually, when we talk about scientific publications, we think about Nature, Science, Journal of Biological Chemistry, these very specialistic publications. Now, the Encyclopedia of Cancer is also known as the Bible of oncology, or the Bible of cancer research, because it has contributions by international authorities across relevant disciplines. So this almost automatically defines us as international authorities in the field of the gene of immortality. If you go to Amazon and you look at the reviews, you see that people who have a very strong competence in the field, like Dr. Paul Clues, a director of the International Agency for the Research of Cancer in Lyon, France, describes the Encyclopedia of Cancer as a landmark achievement in the domain of cancer research, whereas this doctor simply says that this work is really the Bible of cancer. So we're talking about the most prestigious publication in cancer research. And this is not something that was born out of nothing or out of the blue, because this is a research that dates back at least to 1995, so about 21 years ago. About 21 years ago, when I was still working at the University of Florence, Italy, together with my Italian colleagues, I began to study this gene that is called BCL2, that together with other genes that are called master genes, because they do regulate the entire behavior of the cells, we were interested in knowing how these genes were involved in cancer and also in immortality. So these are the first paper that you can find in PubMed, peer-reviewed, that deal with these genes, and then that led us to the current results. Now, what is written in this article in the Encyclopedia of Cancer is a rather scientific and I would say also boring and very specialistic article. So let me try to excerpt a few concepts that may be interesting also for those who are not interested too much in oncology. First of all, we describe the role of this gene called BCL2 in the death of the cells induced by the HIV, the human immunodeficiency virus. So we describe how this gene and the HIV are related and how cancer research can learn from AIDS research and how the topics may help each other, so how AIDS research can help cancer research and vice versa. And then we explore the role of this gene in the physiology of certain cells of our immune system called macrophages that will be described later on, and how these macrophages can be activated by a protein that is called GCMAF, macrophage activating factor, and how all this is related to the function of this gene of immortality and also to a number of diseases ranging from HIV infection to cancer. We also explore another concept that is old and new at the same time. This is the concept of aneuploidy. Aneuploidy, which is again one of those words difficult to be pronounced, means chromosomal disarray. In other words, it was demonstrated many, many years ago at the end of the 19th century by this German scientist, Theodor Boveri, that cancer had some very strong chromosomal instability. Now, this is the chromosomal aberration theory of cancer that went somehow unnoticed for several decades until my mentor, Professor Peter Duesberg of Burkle, of the University of Burkle in California, with whom we wrote together a paper on HIV and AIDS, until Professor Peter Duesberg rediscovered this theory. And again, as you see in this paper published in 2013, it talks about immortality. In this case, is an immortality that we don't like very much because it is immortality of cancer cells, but we will learn that immortality may be a double-edged sword, and of course we want to find the good parts of immortality, not the bad parts. Well, according to the theory of chromosomal instability of cancer, discovered by Theodor Boveri and rediscovered by Peter Duesberg, each cancer is a new species. Like, during the evolution, we have had the evolution of the species. Well, according to this theory that has been widely confirmed and demonstrated, each cancer is nothing else than an experiment of the evolution with the appearance of new species. This paper was published about 5 years ago, and it is a sort of a rhetoric question. Is carcinogenesis, that is the genesis of cancer, a form of speciation? That means the form of the appearance of new species. And also this topic is covered in this article co-authored by myself and John Anderson. But of course, what is most important to the topic of today's talk is the final chapter, the final paragraph of this chapter. What do we write here? Well, let's read it together. In addition to its well-assessed role in oncogenesis, which means in the development of cancer, recent evidence demonstrates that BCL2, this gene, is involved also in physiological processes such as the control of senescence and aging. In the signaling pathway of aging, BCL2 appears to be positioned downstream of telomerase, and I will elaborate on this point later on. Thus, natural compounds showing anti-aging properties upregulate, that is, increase the expression of telomerase, followed by BCL2 expression, for example, in cortical neurons, that are the neuronal cells inside our heads. And therefore, it can be hypothesized that non-neoplastic, that is non-cancer associated, upregulation of BCL2 counteracts aging and may have potential therapeutic roles in the treatment of neurological and psychiatric disorders. Of course, in the Encyclopedia of Cancer, we could not bluntly propose that our research may lead to immortality of human beings. This is the most that you can write in a bona fide scientific article, but let me elaborate on these points. So essentially here we are mentioning telomeres and telomerase. And I think that many of you in your community are very familiar with this topic because we all know that there is a theory of aging that is based on the shortening of telomeres that are here represented in this picture. This is the actual enzyme telomerase, and I'm very confident that you all know Dr. Bill Andrews, who is aging very well, by the way, since he runs the ultramarathon almost every year. Bill Andrews is the one who first identified the gene, the human gene for telomerase, and as he has developed compounds that upregulate telomerase, and therefore they may slow down the process of aging. Now, what does that phrase toward the end of this article mean when we write, in the signaling pathway of aging, BCL2 appears to be positioned downstream of telomerase? I understand that these are very technical terms, but essentially it is a paradigm shift. That is, we are moving from the fight against aging toward immortality. And aging and immortality are absolutely not the same thing. Now, if you go to Wikipedia, many sentences here are taken from Wikipedia just to show that we are talking about common knowledge, and we look for a definition of immortality. It says very clearly, immortality is the ability to live forever or eternal life. Now, this is a kind of a metaphysical concept, but the next sentence is rather interesting. Natural selection has developed potential biological immortality. So now we want to deal with biological immortality as the result of evolution. As you may remember from the papers of Professor Duisberg, taking the old theory of Theodore Boveri, cancer is a form of speciation, the birth of new species, and this follows the rules of evolution. Now we're learning that evolution may even favor immortality of individuals, not only the immortality of cancer cells. Now, according to some definitions, biological immortality refers to a stable or decreased rate of mortality from cellular senescence as a function of chronological age. Now, it is interesting to notice that various unicellular and multicellular species may achieve this state. So immortality is something that does exist in the animal and in the vegetable world, and this state can be achieved throughout the existence or after living long enough. However, and that's important, a biological immortal living being can still die from means other than senescence, such as a strong injury or disease. So let's look at the example of lobsters. Lobsters are very often taken as an example, but unfortunately many things that are believed on lobsters aren't true. Now, all the research suggests that lobsters may not slow down, weaken, or lose fertility with age. And it has been observed that older lobsters may be more fertile than younger lobsters. And it is believed that their longevity may be due to the telomerase. So this is where the research by Dr. Bill becomes very useful, because if lobsters appear not to age, and if this is due to telomerase, that's these are very good news if we can upregulate telomerase. Problem is, however, that contrary to popular belief, lobsters are not immortal. Lobsters, they grow by molting, which requires a lot of energy. And the larger the shell, the more energy is required. And eventually the lobsters, they die. And actually they don't die after a very long time, because the average lifespan for the European lobster is 31 years for males, and as often happens, females live longer, but not that much longer, because their average lifespan is 54 years. Why do they die if they don't age? So here we have a sort of a conundrum. These animals don't age, but still they die. Why do they die? Because of exhaustion during molting. And also, they stop molting, which means that the shell will eventually become damaged, infected, or fall apart, and they die. Now, we want to counteract or to fight this event, because now we have to introduce another complementary theory of aging that goes together with the theory of telomerase or telomeres, and it is the DNA damage theory. Now, the DNA damage theory of aging proposes that aging is a consequence of unrepaired accumulation of naturally occurring DNA damages. And the damage in this context is an alteration of the DNA that has an abnormal structure. Now, nuclear DNA damage, because we know that we have DNA both in the nucleus and in the mitochondria. Now, the damage to the DNA in the nucleus can contribute to aging, either indirectly by increasing apoptosis. Apoptosis means a cell death, or cellular senescence, or directly by increasing cell dysfunction. Now, the damage to our DNA occurs much more often than we usually believe, because in humans and in other mammals, DNA damage occurs frequently, and DNA repair processes have evolved to compensate, but they do not always compensate. In an estimate made for mice, on average, approximately 1,500 to 7,000 DNA lesions occur per hour in each mouse cell, or about 36 to 160,000 per cell per day. So this means that hundreds of times a day, each cell of ours can have a damage inflicted to its DNA. And the accumulation of unrepaired DNA damage is more prevalent in certain types of cells, particularly those that replicate slowly, like the cells in the brain, in the skeletal, or in the cardiac, in the heart muscle. Now, it is also interesting to notice that studies comparing the capacity to repair DNA in different mammalian species have shown that the repair capacity correlates with the lifespan. This means the more a species is able to repair the damage inflicted to DNA, the longer it lives. The initial studies by Hart and Setlow show that the ability of skin fibroblasts, cells of the skin, of 7 mammalian species to perform DNA repair after exposure to a DNA damage agent correlated with the lifespan of the species. And these species were the most common, including the human. In one of the most recent studies, these authors studied a particular enzyme. Also, this enzyme is a molecule difficult to pronounce, the poly-ADP-ribose polymerase, or PARP, which is involved in the repair of the DNA damage. Now, they found that the lifespan of the 13 mammalian species that they had studied, including human cells, correlated with the activity of this enzyme. They went on and they measured the activity of this enzyme in humans who lived 100 years or longer. And they found that humans who lived past 100 years had a significantly higher activity of this enzyme than younger individuals. So now here we are taking into consideration another actor in addition to the telomeres and the telomerase, that is the ability to repair the damage inflicted to the DNA. And if you simply work on the telomerase, you will not age. But if you don't fix somehow the damages to the DNA, you will eventually die, as the lobsters do. So we wanted to work on both fronts. So let's see if these two topics are interrelated, and they are, because this enzyme that repairs DNA, the PARP and the BCL2, are closely interrelated. Now, if we take this article that is extremely recent because it was published only in January 2016, here we see something that may be interesting for those of you who look for natural remedies. In this article, these authors demonstrate how pomegranate extracts, they have a sort of a chemoprevention against the cancer induced in the breast experimentally. And they have this effect through their work on PARP and BCL2. So we see that the two signaling pathways are closely interrelated. But let's see what happens to our brain when the damage at the DNA level accumulates in the cells of our brain. Because again, we want not only to stay young, but we also want our brain to stay young. So we don't want simply to look young. We want to think and to have our neurological functions young as well. Now, if the damage to the DNA in the neurons in the brain occur and they are not repaired efficiently, problems may arise. Because accumulation of DNA damage with age in the mammalian brain has been reported now since 1971, and there are at least 29 studies describing this effect. So again, here the problem is dual. Not only we don't want to age, and this can be achieved working on the telomerase, but we also don't want to accumulate damages at the level of the DNA. Otherwise, not only we die looking young, but also our brain, and as we will see next, our muscles will not function properly. Because the theory of aging due to DNA damage also applies to muscles, both the skeletal muscles or the heart muscle. And as we know, muscle strength and stamina for sustained physical effort have decrements in function with age in humans and all other species. Now, accumulation of DNA damage with age in mammalian muscle has been reported in at least 18 studies since 1971. And this is due to the fact that the protein synthesis and the degradation, the metabolism of proteins, decline with age in skeletal and heart muscle, as would be expected since DNA damage blocks gene transcription. This means that no matter how your nutrition is rich in proteins of high nutritional value, if damage to the level of DNA occurs and if it is not properly repaired, protein synthesis cannot occur and a decline in muscle function will also occur. Now, who or what is the culprit for this DNA damage? Because until now we have been talking about DNA damage, but we have not said what is that causes DNA damage. Of course, nuclear power plants like this one or nuclear weapons may contribute chemicals, toxicants, all these things, of course, can cause damage to the nuclear DNA. But even if you stay away from nuclear weapons, even if you stay away from chemicals, toxicants, and polluted environments, nevertheless, there is always accumulation of reactive oxygen species that cause oxidative stress because of the mitochondrial respiration. In other words, mitochondria are the power plants of our cells. But like all power plants, they have some exhaust fumes, so to speak. And the exhaust are free radicals or reactive oxygen species. Now, the reactive oxygen species, or ROS, they are continuously produced during the life of our cells, whether we are exposed or not to nuclear waste or chemicals or toxicants. And therefore, these reactive oxygen species, they cause DNA damage. DNA damage that, if it is not repaired, will eventually lead to senescence and the death of the cells. Now, the damage that occurs to the DNA leads to a particular type of cell death that is called apoptosis. If you go to PubMed and you look for DNA damage and apoptosis, as I did yesterday, you retrieve more than 28,000 scientific articles. So this means that it is very well accepted that if damage to the DNA occurs, your cells will die. And the apoptosis is a strange term that was introduced in the field of science about 30 some years ago. Excuse me one moment, let me drink a sip of water. And it is a term that derives from Greek, from ancient Greek, and it is pronounced apoptosis, which means physiologic cell death or physiological death. Now, according to the mechanism of apoptosis, when cells feel somehow that a damage to their DNA has occurred, they enter a program that is of physiological death. So essentially they become fragmented, they disappear, and they are metabolized by these cells of the immune system that are called macrophages. So again, the macrophages, they come back into the picture and you will hear about them later on. So to make it very short, when DNA damage accumulates in cells, cells die. And they die through this mechanism that is called apoptosis. Now, since I am 61 years old, as Laurie said, I was born in 1956, of course I'm interested in preventing the death of my cells, because if all my cells die, I also die, and I wish that this happens as late as possible. Now, as it is written here, I think this also comes from Wikipedia, some members of this family of genes that we have described together with John Anderson is in this article in the Encyclopedia of Cancer inhibit apoptosis. That's why they are called the genes of immortality, because they block cell death. But of course, this comes at a price that we will see later on. Now, let's try to see whether it is true that immortality is something that humans may truly achieve. Well, first of all, let's go to PubMed and let's look for immortality human. Well, we find about 800 articles. So this is a topic that has been of interest not only of philosophers, but has been of interest of biologists and medical doctors as well, since there are more than 800 articles published on this topic. Now, going back to peer review papers, we see that immortality, it is at the base of multicellular life, and we can learn lessons from these very small creatures that are called cnidarians. Now let's read only the first line. Cnidarians are phylogenetically basal members of the animal kingdom. It means that they have been with us for more than 600 million years. And together with plants, they share some remarkable features that cannot be found in higher animals. Essentially, they are immortal. Now, what happened during the course of evolution? Why we as higher organisms or as humans have lost this incredible feature of being immortal? Because evidently biological immortality exists. Point is that it does not pertain to us as humans. According to this paper that was published in July of 2014, these authors conclude that higher bilateral, which is a higher organism like humans, have traded immortality for greater complexity and safeguards against unregulated cell growth. This means that during the course of evolution, we have renounced to immortality to achieve a greater complexity and to be safeguarded against unregulated cell growth. Simply means cancer, because cancer is the other face of the other side of the coin of immortality. Excuse me. Cancer cells are immortal, as HeLa cells clearly demonstrate. Point is that we are working on a very thin, on a very fine line here, because we have to decide whether we want immortality at which cost. So let's see if we can reverse this sort of transaction that has occurred during the evolution of species. Now, at least in human terms, every transaction is reversible. So let's see if it is also in biological evolution. That is, if we can have our immortality back without losing the safeguard against unregulated cell growth. This is a rather difficult task, as difficult as finding the Holy Grail. This is an ancient picture, well, it's a tapestry depicting the quest for the Holy Grail. So finding immortality at the same time maintaining a control against unregulated cell growth is truly a difficult quest. And we wonder, where may we find not the Holy Grail, but where may we find mechanisms, products, research leading to this objective? Well, maybe we can find some botanicals in the depth of some alpine lakes, like this one at the border between Switzerland and Germany, so looking for exotic plants. As a matter of fact, there is this botanical that upregulates BCL2 in skeletal muscle. So, as a research scientist, this research was published just a few days ago. In February of 2016, they are looking, for example, for botanicals that could block this death of the cells, working exactly on this gene that we described, BCL2, and this may be useful, if not for achieving immortality, at least to prevent the death of the muscle cells. So we are looking at some rock-solid research, but also some help may come from some other direction in our quest for immortality. Now, probably you recognize Dr. Ilya Mechnikov, who was the first to suggest the possibility of colonizing the gut with beneficial flora more than 100 years ago. So here we're talking about those friendly microbes that are called probiotics. And the first one to use this term was Dr. Mechnikov. Could probiotics help us in achieving immortality, or at least could probiotic inhibit the process of apoptosis, which is the cell death caused by DNA damage? Apparently they can. This paper was published in 2015, and here we see that a very common probiotic, the Lactobacillus rhamnosus, works on the gene BCL2, and this appears to be instrumental in controlling the apoptosis in a small fish that was called Danio, better known as zebrafish. Now you may think, okay, this is the zebrafish. What has the zebrafish to do with these beautiful people attending a convention in San Diego? In other words, what has the zebrafish to do with humans? They are too far away, too separate as organisms, so something that works on the zebrafish may not work in humans. Well, we may be terribly wrong. This is a paper that we have not published as yet. In this paper, that actually was written when I still was at the University of Florence a couple of years ago, and it has a long gestation time. In this paper, we were studying the genes that control human speech. And we were doing a comparative analysis of DNA sequences in mammals and other vertebrates, including the Danio or zebrafish, if you prefer. Now, some very uneducated colleague of mine, when we presented this paper, said, oh, but fish cannot speak. Well, demonstrating all their ignorance about evolution and molecular biology. Of course, fish cannot speak. The point is that they have this gene that serves other purposes in fish. And in this paper, we demonstrate that the amino acid sequence of the Danio in this segment shows a large homology with the human sequence. In other words, the zebrafish has many similarities in its DNA, in its protein sequences with humans. And therefore, we cannot infer, we don't have the demonstration, that an approach such as that described before might work in humans as well. However, the upregulation of BCL2 may pose a serious challenge, because let's not forget that BCL2 stands for B-cell lymphoma, number 2. In other words, this gene is typically upregulated, that is, its expression is increased in cancer cells. So here it is a little bit like the story of telomerase. Telomerase is highly active in cancer cells, so at first you may wonder, do we really want to increase the activity of telomerase? And the same applies to BCL2. BCL2 is highly active in cancer cells and makes cancer cells immortal. So you may wonder, do we really want to increase its activity? Well, we are walking on a thin ice, but essentially, yes, we want. Because when BCL2 is hyperactive, as it happens in cancer cells, it is because of translocation. That is, that chromosomal aberration that was first discovered by Theodor Boveri and then rediscovered by Peter Duesberg. So this is what we do not want to happen. We do not want mutations. We do not want chromosomal aberrations or translocations that hyperactivate BCL2, because cells will become immortal, this is true, but they will be cancer cells and we don't want them. However, we may want to upregulate, that is to increase the activity of BCL2 in healthy cells or in cells that are aging. But of course, if we want to do this, we need some form of defense. Actually, what we need are good sentinels like those. All right, so we need sentinels. And the sentinels in our body are these cells of the immune system that are called macrophages. I began working on macrophages 26 years ago when I published this paper in PNS in 1990 describing the effects of a protein that is called macrophage colony stimulating factor on the activation of macrophages. Now, let me show you what happens when a human breast cancer cell, that you can see a different level of magnification, the human breast cancer cell is this one, then is this one, then is this one, and now is this big one. Is always the same, but these are different levels of magnification, is attacked in a search and destroy mission by activated macrophages. In this case, macrophages activated by this protein called GCMF. Now we have evolutions of this. Now, essentially, the macrophages, they are able to recognize the cancer cells, they attack them, so these little dots are macrophages, this big cell is a cancer cell, and they induce the cell death, this is the nucleus of the cancer cell, by fragmenting the DNA. So the DNA of the cancer cell is all fragmented, so this cancer cell is undergoing a process of apoptosis because all these friendly sentinels that are our macrophages, they are recognizing, attacking, and killing the cancer cell. So if now we are able to empower the macrophages so that they can attack and kill the cancer cells, we shouldn't be afraid of upregulating, that is increasing the activity of the BCL2 gene that will guarantee, if not our immortality, definitely will slow down, together with telomerase, the process of aging at the cellular level. This is a further magnification. Here you can see the macrophages truly excavating the cytoplasm of the cancer cell and inducing the fragmentation of the DNA. However, sentinels may not be enough. You have to give them weapons. You have to give them instruments so that they can recognize their enemies. By the way, if you wonder where all these backgrounds come from, at least many of them, they come from a place in southern France that is called Arles. Arles is where the original Champs-Élysées or Alice camps were founded. So it's a burial site, very, very old, hundreds, if not thousands of years old. And I thought it was sort of appropriate when we're talking about fighting death and looking for immortality. Now, sentinels may not be enough. We also need other things. And a class of compounds that have recently gained the attention of the researchers are the protease inhibitors. Now, these are molecules that have many functions in fields as diverse as medicine and agriculture. And actually, this is a field where I began to work in the early 80s when I was studying the relationship between cancer and nutrition. And this is the very first paper that I published when I was 24 years old. It is written in Italian, but it is still retrievable in the Department of Agriculture of the US, in the National Agricultural Library, and it explores the correlation between food and cancer in this area of Northern Africa. And I was working on protease inhibitors when, as Laurie said during her beautiful introduction of my work, I published this paper sponsored by Sir John Vane. This is Sir John Vane, Nobel laureate in the 70s. And this paper actually describes protease, and in this case also cyclooxygenase inhibitors. So these are molecules that I'm very familiar with. And just a few days ago, I would say at this point, a few hours ago, in March 2016, this paper was published demonstrating the potential of these compounds to reduce the induction of carcinogenesis, and the keywords for this paper are rather odd because they mix agriculture with Alzheimer's disease, cancer, cardiovascular disease, HIV, plant pathology. This is simply to show the extent of the usefulness of these compounds in fields as diverse as agriculture and medicine. Now, we are still looking for the holy grail. That is, we are still looking for the compound or the compounds or the tools or the mechanism that will allow us to upregulate BCL2 and therefore preventing cell death at the same time avoiding cancer. According to some, one of the holy grails could be found in Valencia, Spain. I was there a couple of years ago for a congress. I don't know whether it is the original one or not. So where do we find solution? Obviously, we find solution in the field of nature, looking at the things of nature, or as my good friend in Milano, Dora, would have said, looking at the nature of the things. And by the way, I would thank her for teaching me some Latin, this is a text in Latin. So looking at the things of nature, we may find the answer to this quest for immortality that passes through our study of the BCL2 immortality gene. But now, for the last 5-10 minutes of my talk, let's change topic. Why? Because until now, all the times I've been talking about immortality, I've been talking about cell death, DNA damage, nuclear DNA, and so on. I was only referring to the 10% of my body. In other words, it is now well assessed that we are only 10% human. We're only 10% human because for every human cell in our body, there are 10 microbial cells. In other words, our body in this very moment is composed by a certain number of human cells and by 10 times more microbial cells. And if we look at genes, the ratio is even worse. The microbial genes, that is the information, the life information in our body right now, the microbial genes are 100 times more than the human genes. Human genes are about 22,000. Microbial genes have not been counted, but it is estimated that they range between 2 millions to 8 millions. So at worst, there are 100 times more microbial genes than human genes. So until now, I've been talking about the concept of immortality of our human cells. But what happens to the microbial part of our body that, as we now know, constitutes the major part of the information? As you may know, about 1 year ago, together with my good friend, Peter Greenlaw, we published this paper, Your Third Brain, where we postulate that the microbiome may function as a third brain. Now, we will soon write a new version of this paper, because of this book, because now we know that we may have even a fourth brain, but this is another topic. So what happens to this other part of our body? Well, in a book that yet has to be published, I wrote a chapter that it is entitled, Your Third Brain Will Live Forever. So this is another concept of immortality. In other words, immortality of human cells is one thing, but we also have to deal with the concept of immortality of the microbial cells that form such a substantial part of our body. Now, in this chapter of this book that will be out, I hope, in a few months, I explore the philosophical paradox of the ship of Theseus. So it's a very complex topic that has fascinated the philosophers for thousands of years. You can find some news about it in Wikipedia. The point is that we have to find out whether our microbiome retains consciousness of ourselves, because the microbiome will definitely outlive our human cells, no matter how strong is our search for immortality. So we have to explore these concepts as well. Now, I have been speaking for about 1 hour, and as I said before we began this talk, it is well known to neurologists that the human span for attention is 45 minutes. So I think it is for me time to stop and wonder whether this is the end, Now, I'm showing here a paper that was published by Kurt Gödel in 1949. It's an extremely complicated paper that few people in the world can understand, probably not even Einstein, to whom the paper was dedicated, could understand it in its depth. But to make it very, very short, according to this theory, there is an eternal recursion. So most likely we will find ourselves here again and again. I don't know whether this will solve our quest for immortality, but definitely it has been a wonderful time to spend with you. I hope that I have been able to convey my message. So I stop it here and I wait for your questions.
Wow. Thank you so much. I know I have a ton of questions, but I want to open up the lines, and for anyone who has either clarification, because Dr. Ruggiero covered a lot of information. If you have questions about where this is leading, perhaps implications in terms of everyday life, I know that's what's going on in my brain right now, just unmute your line. Everybody's currently muted, so the bottom left side of your screen, you can unmute and just say your name and any question or feedback or information you'd like clarification on.
This is Lori. I have a question. Dr. Ruggiero, you were talking about at one point about the GCMAF activating and inducing cell death of the cancer DNA, and then you talked about the BCL2 gene. So, and the question you put was, how do you increase, talking about the increasing the activity of the BCL2 gene without inducing cancer? And I kind of got lost there.
Well, you are lost because this is the most difficult task. So you're not the only one of being lost over there. To make things very, very simple, first of all, the BCL2 gene was found because it is overexpressed. It means it is hyperactive in cancer cells. In cancer cells that are immortal. So this is why the gene was defined the gene of immortality, because it makes cancer cells immortal. And this in itself is not a good thing at all. However, as all the other genes, it also serves a function in healthy cells. There are not genes that work only in cancer cells. All genes, they have a function in healthy cells, and this function then it is exploited by the cancer cells for their own sake. So scientists began to wonder, what is the function of the BCL2 gene in normal cells? And they found that its function is to allow the cells to live longer so that it did not die prematurely. So it is a gene that serves a purpose in healthy cells. Also, these genes is important because it gives time to the cells to repair the damage inflicted to the DNA by a number of factors. First of all, the reactive oxygen species. So the point now is, if I find a compound, and there are many, that upregulates, that is, increases the activity of the BCL2, I'm doing a favor to the healthy cells, no doubt about this, because I will prevent their death. I will give them time to repair the damage inflicted to the DNA, and at least theoretically, they may live forever. But at the same time, I'm doing a favor also to the cancer cells. Cancer cells, who, by the way, may not even need this favor of mine because they have already found the way to activate the BCL2 gene through chromosomal translocation. So if I want to do this, I have to bear in mind that at any time in my life, not only now that I'm 60, but also when I was 2 years old, there are cancer cells in my body. And this has been demonstrated over and over again. So we always have cancer cells in our body. However, as long as our immune system is able to keep them under control, cancer does not arise as a disease. If you have a couple of cancer cells somewhere in your body, they don't bother you. But if you have millions of cancer cells all lumped together, then it's a problem. So essentially, if I want to increase the activity of the BCL2 gene, because this does a favor to the healthy cells, keeps them healthy, gives them time to repair the DNA damage. At the same time, I have to empower the immune system so that the immune system can go on a search and destroy mission to find the cancer cells and kill them before they organize under the form of a mass. Now, there are many, many ways to empower the cancer cells. Activation of macrophages is one way, and it is a very promising way. Of course, it's not the only one. So the point is that if we want to increase longevity, at the same time, we have to work in empowering the immune system. Otherwise, we run the risk of doing a favor to the cancer cells that are always in our body. Now, according to the theory of aneuploidy that I mentioned at the beginning of my talk, cancer cells are like alien species. That is, they follow the rules of evolution. They fight for their life, and they have all the tools to fight, and they fight well, unfortunately. So we have to have better fighters. We have a wonderful and excellent immune system, but also the immune system suffers the damage caused to the DNA by the reactive oxygen species, by the chemicals, the toxicants, all the things that you want to get rid of when you detoxify, and so on. So in other words, the bottom line is, we can achieve longevity. Hopefully we can achieve something close to mortality as other organisms they do, but only if we work on both fronts. That is, working on the mechanism that guarantee longevity and slows down aging, at the same time empowering the immune system so that these sentinels of ours always look for and destroy the cancer cells. As I said at the beginning, immortality is a double-edged sword, because immortal cells, they do exist, don't wonder. They do exist very well. They're called cancer cells, but we don't want that kind of immortality. I don't know if I've been able to explain this topic, which is very complex.
That's much clearer. Thank you.
I have a question, actually, or a comment. Is the research being done, or is the goal to try and block BCL2 from entering cancer cells in order to maybe cure cancer or prevent it from the cells from being stronger or getting stronger? Is that the idea, to try and block that from getting into cancer cells?
Exactly. So there is a lot of research going on BCL2 in the field of molecular oncology. To be more precise, is not a problem of the BCL2 to enter the cancer cells, because BCL2 is already inside each cell, not only the cancer cells. Problem is to avoid those chromosomal aberrations that lead to the hyperactivity of the BCL2 gene in cancer cells. Now, here we are facing exactly the opposite problem that we were talking just 1 minute earlier. In other words, we have many inhibitors of BCL2. They do exist, and they are used, or they are working on research to use them as chemotherapeutic agents. Because of course, if you block BCL2, you block immortality, and you block immortality of cancer cells. And this is something you may want to achieve. You pay a price, that you kill also the healthy cells. So it is like in many other instances in chemotherapy. Most chemotherapeutic agents work. Problem is that they have side effects. Why? Because if they work in killing the cancer cells, most often they also kill the healthy cells. In the case of the inhibitors of BCL2, we are facing exactly this problem. You stop the immortality of the cancer cells, but you also favor the death or apoptosis of the healthy cells. Again, it's a very fine balance. So that's why the field of immunotherapy is so promising in these past few years, because in immunotherapy, in particular in the immunotherapy of cancer, what you are doing is not to try to kill the cancer cells with some exogenous drug, but it is to empower the immune system, so it is the immune system that goes and kills the cancer cells. At least in theory, immunotherapy is devoid of side effects. And then, of course, there are many forms of immunotherapy. Some of them are more advanced, some of them are more in the phase of research. But yes, your point is very well taken, and again, is a very fine line. Immortality, we want immortality for healthy cells. We want to deprive cancer cells of their immortality.
So I guess for everybody who's listening tonight, I guess the message for us is that we're all united in trying to bring more health to the average person out there through the products that we're involved with, nutrition systems we're involved with, which will strengthen the immune system, which will help to fight off the cancer cells. That's the bottom line, I think, for a lot of us.
I think that you're absolutely right, and it has been very well demonstrated that what is labeled as healthy lifestyle, which of course is centered on nutrition, is the best way to prevent the onset of cancer, but it is also the best way to fight cancer together with conventional therapies. Let me just, this is a little bit off topic, but let me just tell you that in the fight against cancer, nutrition is paramount. Nutrition is paramount in preventing a cancer, but also if you have a cancer, nutrition is paramount in enabling every type of therapy to work at its best. So this has been demonstrated for radiotherapy, for several types of chemotherapy, for several types of other approaches. So if nutrition is taken care of, all therapies will have a good probability of having a success. But if nutrition is not taken care of, all therapies, even the most promising, probably will fail. So I think everybody fully agrees with your concept that a healthy lifestyle based mainly, even though not uniquely, on nutrition is absolutely the best way to prevent cancer, in the complementary therapy of cancer and in many other diseases, and of course, if you want to live longer, healthier lives.
I'll mute now, turn it over, but thank you. I got a tremendous amount out of this call for sure.
Thank you.
One of the questions I had, Dr. Ruggiero, it's Lori, is in terms of, I'm fascinated with the third brain, and for years have been looking into that. And when you show that diagram that we really are only 10% human, the role, and more research is being done that our food doesn't have the healthy microbes that are required to keep our body working the way it should. So in terms of how that plays out in our healthy immune system, current research is saying supplement with a proper probiotic, for your gut health. And in terms of how that works with what you just talked about with those microbes, is that what you're referring to, the prebiotic, probiotic ratio that needs to be present in the gut in order for the rest of the body to function properly?
Yes, well, that's a very complicated topic, so let me try to explain in just a few minutes. First of all, it is now well accepted that the major organ of the immune system is the gut, but not the gut meant as a digestive tract. The gut because it is where the largest part of the microbes, of the microbiome reside. So the microbes, the microbiome or the intestinal flora, microflora, different names to indicate the same thing, is an essential part of the immune system. Let's take mice with a perfectly efficient immune system. Let's sterilize their gut with powerful antibiotics. They become immune deficient as if they had last stage AIDS. Now let's give them back their microbes that of course are different than human microbes. their immune system begins to work exactly as it was before. So there are now hundreds of publications demonstrating that every type of imbalancement of our gut microflora leads to some malfunctioning of the immune system. This is extremely well accepted. Now, when it comes to the topic of probiotics, things are a little bit murky, because with the term probiotics, people may indicate very, very different things. Now, the term probiotics was introduced simply to indicate that not all microbes are bad, because until a few decades ago, when you said the word microbes, everybody was terrified because of the infectious diseases. Now we realize that, of course, there are pathogenic microbes, and we want to get rid of those or to prevent the infection by those, but also there are some very friendly microbes that actually are part of our body. And these friendly microbes have been termed probiotics. Now, I have some expertise on probiotics, and let me share with you some information. Probiotics, they work at their best when they are in their natural environment, that is, in fermented foods or drinks. So let me share with you this information that now it's about 6 years old. One of the major issues in immunology, of course, is HIV infection and AIDS. By definition, AIDS is severe immune deficiency. And as you know, AIDS is a problem in poor countries in the world, in particularly in Africa, because it is associated with malnutrition, with a number of other issues. So about 6 or 7 years ago, some of the best microbiologists in the world, coming from Canada, a research group led by Dr. Reid, they went to Tanzania, which is one of the poorest countries in Africa, in one of the poorest neighborhoods of Tanzania, where there is a high incidence of HIV and AIDS. They brought with them some ferments to prepare a probiotic yogurt. They taught local women to prepare a probiotic yogurt using local cow milk that they could find over there. And so these communities, they began preparing this probiotic yogurt. And they began giving the probiotic yogurt under the form of a real yogurt to a number of people attending their community. After a few months, they observed a significant improvement in the function of the immune system of these people living with HIV and AIDS. So all the parameters that are typical of HIV and AIDS were improved by the consumption of the probiotic yogurt. And they were very happy. They had found a rather inexpensive and simple way to provide this population with a food that is nothing but a probiotic yogurt that was helpful in restoring the immune system. And they published a paper in the Journal of Gastroenterology in 2010. Wonderful. Now people began to criticize and say, okay, yes, your experience is wonderful, but a probiotic yogurt can be delivered only to a very limited amount of people, only the people who can attend that community, that ambulatory or whatever it was. Definitely it cannot be shipped because it should be shipped refrigerated, and this is rather impossible in Africa, but also in the Western world becomes complicated. So they said, okay, your observation is good, but cannot be applied on large scale to help people infected with HIV or having AIDS. No problem, they say, we are microbiologists, so we can devise a system, so we encapsulate the same microbes into some capsules that can be stored in shelves, can be shipped anywhere, don't require refrigeration. You can even drop by parachute in the middle of the jungle if you wish. So essentially what they did was to take absolutely the same strains of bacteria that had proven so effective in the probiotic yogurt and put them in a capsule. Of course, since they are very respectable scientists, first they ran an experiment. And what they observed was that if they gave the encapsulated probiotics instead of the yogurt as a common yogurt, the effects of the immune system were zero, nothing. And they concluded that this striking difference was due to the fact that in the natural fermented food, like a probiotic yogurt, the microbes are together with their environment that they have contributed to create during the process of fermentation. However, in the encapsulated probiotics, the microbes were by themselves. And even though they were alive, they were not able to restore the immune system. So this is why when we're talking about probiotics, we're talking about a very difficult issue. But in general terms, fermented foods are much better than encapsulated probiotics. This is a general statement. Of course, again, don't take this as a medical advice, but just in general terms, fermented foods, and you find many types of fermented foods. You find the kefirs, you find yogurts, you find kombucha, you find a number of sauerkraut and so on. So several types of fermented food products have been developed in a number of different cultures all over the world, and they are truly beneficial. Encapsulated probiotics is a rather different issue.
Wow. Thank you. That was, I didn't realize that at all. I thought the pill form was equal. So knowing that the fermented version makes sense. Thank you very much. And I think what we'll do at this point is definitely take Dr. Ruggiero up on his offer at the beginning of this evening's program to have another session, because I know that there are many, many topics that I would love to learn more about. Just, I could imagine another presentation just on the fact that we are really only 10% human. And I think just that one topic alone, I would love to hear more about. So presentation, Dr. Ruggiero, it is an absolute thrill and a joy to be able to learn more and understand more about the miracle of the human body. And you make the science and the research so easy to understand and so simple and really so exciting as well. I mean, the cusp of so many amazing opportunities for life and for extending life in a really powerful, positive way is one of the biggest takeaways from this evening. So once again, thank you so much for your gift of your time, your knowledge, your expertise, and thank you for everyone for the incredible questions that you wrapped up this evening with.
Thank you, Lori. I really appreciate your help and your support, and we will see you next week or whenever it is convenient for you. Thank you.
Good night, everybody.
Bye-bye.